polypeptide synthesis, or synthesis of proteins. Hello to everyone watching this video. Today we'll be taking a look at how proteins are made and why they are so important to us. To start off, let's take a look at whereabouts DNA is found in the prokaryotic and eukaryotic cells. The DNA of eukaryotes and prokaryotes are chemically the same, but differs in how it is structurally arranged into chromosomes and packaged inside the cells. As you can see, prokaryotic cells have a much simpler structure than eukaryotic cells. They contain a single chromosome in the form of a circular strand of DNA. This is a prokaryotic cell. It has DNA in the plasmid and the chromosomes. This is a eukaryotic cell. The DNA is located in the nucleus within the cell. Individual DNA molecules are arranged into a number of separate chromosomes. Proteins are so important. They have so many functions. To name a few, they create antibodies, messengers, enzymes, and structural components. Number 1. Antibodies. Antibodies bind to specific foreign particles such as bacteria and viruses to help protect the body. Number 2. Messengers. Messenger proteins, for example some hormones, transmit signals to coordinate biological processes between different tissues, organs, and cells. Number 3. Enzymes. Enzymes carry out the thousands of chemical reactions that take place in the cells. By reading the genetic information stored in DNA, enzymes also assist with the formation of new molecules. Number 4. Structural components. Structural component proteins provide support and structure for cells, and looking on a larger scale, structural component proteins also allow the body to move. Now back to making proteins. The process of creating proteins starts off with a cell. Taking a look inside the cell, we see the nucleus. The nucleus contains the genome. In our human bodies, there are 23 chromosomes split up in the genome. Each and every chromosome contains a long strand of DNA tightly wrapped around proteins called histones. DNA consists of long chains of nucleotides wound into a double helix. Within the DNA, it is sections called genes. These genes hold the instructions for making protein. Transcription begins when an enzyme called RNA polymerase connects to the start of the gene. The RNA polymerase then unzips the DNA. Only one strand of the DNA contains the genetic information to make proteins. It is called the non-coding strand or sense strand. The sense strand acts as a template and RNA nucleotides are assembled, which are bonded together to form a complementary single strand of mRNA. The M in mRNA stands for messenger. The sequence of the nucleotide bases on the mRNA molecule is the same as the DNA coding strand, except that it has a U instead of a T. There is usually a significant amount of mRNA editing to be done, which is critical for the process to work properly. This whole process is called transcription. The mRNA then moves out of the nucleus through a nuclear pore and into the cytoplasm, where it then encounters some of the million ribosines in the cell. Usually, one mRNA molecule is read by a large number of ribosomes, so multiple chains of the same polypeptide products are produced from one mRNA template molecule. Now begins the process of translation. Translation occurs when the ribosome moves along the mRNA molecule, and as they do, they attach tRNA molecules to the mRNA by temporarily pairing the bases of the tRNA anticodons with their complementary triplets of bases, which are codons, on the mRNA. On the tail end of each tRNA is one of the 20 different amino acids which are linked to one another by an enzyme to form a polypeptide chain. Each amino acid is then sliced off its tRNA carrier. The tRNAs move away from the mRNA, leaving the growing chain of amino acids and move back into the cytoplasm where they can pick up another amino acid and be reused. When the last amino acid has been added, the polypeptide chain folds into a complex 3D shape, forming the final protein end product, which can be one of four things: primary structure, secondary structure, tertiary structure, and quaternary protein structure. Number 1. The primary structure. The primary structure is the basic amino acid chain that is folded into a protein. Number 2. The secondary structure. The secondary structure is a three-dimensional arrangement of the polypeptide chain as a result of the amino acid chain becoming linked by hydrogen bonds, either twisting the polypeptide into a spiral or folding into a pleated sheet. Number 3. Tertiary structure. Tertiary structure is further folding from the secondary structure. And number 4. The quaternary structure. The quaternary protein structure occurs in proteins that are made up of two or more polypeptide chains that are linked together to create a more complex three-dimensional structure. This is a table of the mRNA codons. On the mRNA codon table, there are three stop codons: UAA, UAG, and UGA. The codon AUG is the start codon, signifying where translation should be initiated. There is more than one codon that codes for each amino acid, creating some flexibility for errors in the genetic code. This concludes polypeptide synthesis, or synthesis of proteins. Thanks for watching.